Good morning students. Hope you be fine at your respective places. Beta. Today we are going to discuss one more topic this regarding this process of photosynthesis, and we will come towards this light because we know that this light, that's the sunlight, it is the very important what we call the raw material for the process of this photosynthesis. Now, how we can define the light? Light can be defined as the form of energy. Light can be defined as form of energy, and this light, अगर हम बात कर लेंगे, it travels in the form of stream of tiny particles. So what we have studied, that light is the form of energy, and this light, it travels in the stream of tiny particles. Now we know, we have already discussed what we have discussed, that जो हमारे पास light का एक feature जो हमको पढ़ते हैं, the various factors regarding this process of photosynthesis, of which light के मुतालिक अगर हम पढ़ लेंगे, we have one factor, that is the quality. That factor is the quality. Now, how we can define the quality? यहाँ पर we can say that which wavelength is essential. Which wavelength is essential? Which wavelength is important? Which wavelength is essential for because we say we have what we call as studied all the different types of these these pigment. We have discussed the chlorophyll, we have discussed the carotenoids, we have discussed the phycobilins, and we know that different these pigments, they absorb different wavelengths. Now when we discuss like this, what we will in this, that we get the one more term that is called as the absorption spectrum. Now what do we mean by this absorption spectrum? It is rather the what we called as the curve which represents the light absorbed. This what we call as this lights in this absorption spectrum, it is simply the curve which represents the light being absorbed at each wavelength by pigment. Okay, a pigment, it absorbs the light at different wavelengths, and when we draw the graph, we get the curve. That curve is called as the absorption spectrum. Then we have got one term that is called as action spectrum, for which color अगर हम बात कर लेंगे, जो this pigment जो है कि we have got different colors of the light, for which color अगर हम देखेंगे जो action जो है that is maximum. There we got this graph. That graph is called as the action spectrum. So let us draw the graph. We have got different what we call as this pigments. For example, this is chlorophyll A by this green color. Then with this I have got one more, that is chlorophyll B, that is with this black color. chlorophyll B. Then we have got one more, that is carotenoids. Let it be with red color. Then we have got phycobilins. Let it be with blue color. Now draw the graph. Draw the graph. Now wavelength है, हम यहाँ से start कर लेंगे 400 nanometer from this nanometer. Then this is 500 nanometer. Then we have got one more. This is 600 nanometer. Then we have got 700 nanometer. Now we know that light is the mixture of different colors. For example, we've got violet, indigo, blue, green, yellow, orange, and red. See what happens if we draw the graph for chlorophyll A. For this chlorophyll A, जो हमारे पास की है, we have shown it with A. It is green color. So when we draw the graph, we will see the action is maximum. What we will see, the action is maximum at blue and at red. This is chlorophyll A. So when we draw the graph, we will see that this action जो है, we will find the two maximum peaks, and these peaks are found at the blue color and one at the red color. Now for the chlorophyll B, again I will draw it with this. Again you will see the maximum, it is again with this, like this, maximum with the blue color and another with this red color. Then experimentally proved. Then when we draw the curve for this carotenoids we have seen with this, what we call as a red color. What we will see for the carotenoids, if we draw the graph, we will see for this green color, this is again. If we see for the phycobilins, again what we will see, like this. So we will see this absorption spectrum. It is shown by this absorption spectrum. It is shown by means of, it is studied by means of this एक apparatus हमारे पास है, that is called as spectrophotometer. So for this absorption spectrum, it is studied by means of the apparatus that is called as spectrophotometer. And while studying this graph, what we will see that the absorption spectrum जो हम यहाँ पर देखते हैं of chlorophyll A and chlorophyll B indicate that these pigments, they only absorb blue and red lights. And what we will see, जो action spectrum, they show that the maximum photosynthesis they mainly takes place at the blue and what we call as the red color. Now one more experiment was there, and that experiment, उसमें हम क्या देखते हैं, what we will see in that experiment. See, this is the trough, and in this trough we have put this alga, green alga is there, like this. I will dot this. This is the green alga, this is the green alga, like this. Again I will draw this violet, indigo, blue, green, yellow, orange and red. When we will see that sunlight, what we will see, this is in the water, this is in the water, this alga, it is in water. What we will see in the red spectrum, what we will see at blue, what we will see, like this, air bubbles are formed at this place. Air bubbles are formed at blue, this red. What we will see, bubbles are formed. After this experiment, what we will see, this bubble जो है, they are mainly the, what we will see, these bubbles, they are mainly जो वहां पर bubble, it is because of the what we call as the oxygen gas O2, which has जो वहां पर हमको रिलीज होता है, जो वहां पर हमको बन जाता है. The oxygen gas is produced when this alga, it is illuminated in the light. This light is the mixture of 7 colors. At blue light, what we will see, that there are these air bubbles, and at red again we find the air bubbles. And when we test these air bubbles, we found that these air bubbles, they are the oxygen gas which has been produced. So it indicates that during the process of the whole process of photosynthesis has taken place. So this whole process of photosynthesis, that is maximum, जो हम देखते हैं एक्शन जो है, एक्शन स्पेक्ट्रम, दैट इज मैक्सिमम इन द ब्लू लाइट and that is maximum in the red light, because at this case what we will see, these bubbles are formed and the oxygen, these are bubbles, they indicate that the oxygen gas is produced. So when we see the whole process here, we will see when the reaction हम देखते हैं photosynthesis की, that oxygen gas is formed during the process of photosynthesis. So जो एक्शन स्पेक्ट्रम हम देखते हैं, वट एवर वी विल से एब्जॉर्प्शन स्पेक्ट्रम हमने पढ़ा, एक्शन स्पेक्ट्रम ने देखा, हियर वी विल सी जो मैक्सिमम हमारे पास व्हेन द algae it absorbs the light, and when we will see the action हमने देखा that in the blue light maximum of these air bubbles are formed, and in the red wavelength here we will see that this bubbles are formed, which indicates that the oxygen is produced. So by these experiments, it has been proved that the rate of photosynthesis is enhanced. It increases in the blue wavelength and in the blue light and in the red light. So we will see the action spectrum shows that the maximum photosynthesis, it takes place in the blue and red region of the spectrum. Actual हम क्या देखते हैं कि that in terms of the oxygen, हम ये देखते हैं कि chlorophyll A and chlorophyll B, they show the maximum. This maximum हम क्या देखते हैं कि show the maximum absorption in the blue and the red region. This was about this light absorption and what nature जो हम देखते हैं different wavelengths, how they play an important role in the process of photosynthesis. So this was all about today's topic. In the next video we will proceed further. Until then, goodbye. God bless you. Thanks.